As a reminder, you should not be starting this video immediately following the fact that you've watched 3.1. You need to have done the Socrative questions that review the functional groups. You need to watch Amoeba Sisters to introduce you to this concept first, and you need to be working with your textbook to let you sit down and work through these material, because we're just going to go through the basics here pretty quickly, and it's important that you have a good foundation to start off with. So there's going to be a few additional review questions. Please go ahead and go into Socrative at this point and do these 6 review questions, and then come back. So a molecule's shape determines its function. Big theme, keep this going, don't forget it. If you want to think about the function of a molecule, you always have to hark back to what shape allows it to have this function. Now, why are living organisms carbon-based? It's all of the above. Carbon forms highly stable covalent bonds. It can form 4 of these bonds, and it can form bonds with many other molecules. So this is a diverse component. And these functional groups, when added to the diversity of our carbon backbones, give additional notable and important properties. Plus, they're chemically reactive, which is important for us being able to synthesize larger components. Now, which functional group will make the molecule acidic? That's carboxyl. By the way, which functional group would make it basic? That's an amino group. Now, name the reaction that forms bonds between monomers. That would be a dehydration synthesis. Synthesis, to synthesize, to make something. So a dehydration synthesis is always making a bond. The reverse is hydrolysis, or lysis means to break. So hydrolysis is the breaking of the bond. Now, what molecule is released from the formation of this new bond? This is a dehydration synthesis, meaning the removal of water, because we are removing one molecule of water. So I want you guys to continually be thinking back to 3.1. Think about functional groups, think about structures that this gives the macromolecule, because that's the core behind properties and functions. And I do want you to be prepared, because you do need to know this material in order to assemble an actual cell. So we're going to start with a discussion of proteins, because proteins are the most abundant macromolecule in the cell. They actually make up about 50% of the actual mass of your cells, at least once you were to remove the water. Now, an important type of protein is an enzyme, but there's a theme in this class. We're going to look at proteins over and over and over again, and we're going to really try to better understand the fact that because proteins can form so many different functions, they can form so many different structures, that they can form so many different functions. So the fact that proteins are extremely abundant is in part because of their diversity allows them to do so many different things. The first one that I want you to know is enzymes. And we're going to see the fact that these enzymes, these proteins that act as enzymes, will help to speed up dehydration synthesis reactions necessary for the creation of these macromolecules. Other functions can include transmembrane transporters, receptor proteins, muscle contractile proteins, so on and so forth. There's a nice little review of these in your textbook, and I would recommend that you pay attention to these if you haven't done so before. Now, the backbone of these proteins are going to be amino acids. Amino acids are the monomer formed of an amino group and a carboxyl group. That carboxyl group is acidic, so these are called amino acids. There's also a central carbon, and from that central carbon extends a variable group, meaning it's different between different monomers, different versions of amino acids, sometimes called a side chain. This R group is going to be what is actually responsible for giving the property to that amino acid. There are 20 different amino acids. Each has the amino group, central carbon, and carboxyl group. The only difference between them is going to be that side chain. And these side chains provide properties including becoming hydrophobic, hydrophilic, or going to even be so far as acidic or basic. In order for you to determine which one of these amino acids you're dealing with, you need to know a few things. The first is the basic structure of an amino acid. Because every amino acid has an amino group, a central carbon, and a carboxyl group, we do not consider this when we look at differences between amino acids. So what you need to do is cross off that component of the molecule. Then what you can do is identify the side chain. And once you've identified the side chain, this will allow you to take a deeper look at the molecules that compose that side chain. You're looking for specific functional groups. If you have only non-polar covalent bonds, then that side chain is going to have a hydrophobic nature to it. Even though the backbone of the amino acid is still going to be a basic, acidic, hydrophilic, the actual side group itself is going to be responsible for the interactions that that amino acid has in the larger context. So we label that as a hydrophobic amino acid. Hydrophilic amino acids are going to be things that have polar covalent bonds. Basic are going to have basic functional groups, such as an amino group, not the amino group that's at the core. It's going to have an amino group in its side chain. And then there's going to be acidic side chains. And these are going to have acidic functional groups. Once again, not the acidic carboxyl that's at the core of the amino acid, but an acidic group within the side chain. So now I want you to try and identify the type of amino acid that leucine is. So in order to do this, you need to start by identifying the core structure of an amino acid. Once you've done that, you can cross off that core structure. That allows you to identify the side chain, and then you need to look at the components of that side chain. So go ahead and tell me your answer in Socrative, and then come back when you're done to see how you did. Hopefully you were able to recognize that this molecule is upside down from your previous figures. Our side chain is at the bottom. You'll see on the left is our amino group, NH2. On the right is our acidic carboxyl group, COOH. So that's the core structure of the amino acid. So our R group here is composed entirely of carbons and hydrogens. So this hydrocarbon is going to be nonpolar, which means that this is a hydrophobic amino acid. When you connect amino acids together, the bond between them is known as a peptide bond. So we call long chains of amino acids polypeptide chains. The enzyme that is responsible for synthesizing the peptide bond is a ribosome. Ribosomes are going to be responsible for forming proteins. And since the bond that holds the monomers of proteins together is known as a peptide bond, forming these polypeptide chains, ribosomes are responsible for that synthesis. Now, when we take a look at these peptide chains, these polypeptides, what we're going to notice is that there's always a connection between one carboxyl group and one amino group. Since that leaves one end of the chain to always have an amino group, we call that end of the chain an N-terminus. N for the nitrogen at the core of the amino group. The other end of a polypeptide chain will always have a carboxyl group, so we call this the C-terminus. When we get to our genetics section of the class, you're going to be synthesizing these chains. You're going to be recognizing which amino acid and in which sequence, and you're going to have to label the ends of that chain. So it's going to be important that you understand what N-terminus and C-terminus mean and how to determine which end you're dealing with. Polypeptides are going to be a string of amino acids that are placed together. And the order of the amino acids is what's determined by your genetics. We're going to have a whole section on that. But it's important to note that these polypeptide chains don't fold into their final molecular shape randomly. That structure is a very specific interaction that we call the level of structure. So there's going to be multiple interactions that are occurring that actually result in the shapes that you see. And so therefore, the levels of structure that determine that final protein shape are really what's responsible for determining its function. So we're going to go through those 4 levels of structures now. The first level is primary structure, and it is simply going to be the order of amino acids that are catalyzed by the ribosome. So when we choose to put a peptide bond between 1, 2, 3, and 4, that order of those amino acids is what we know as primary structure. This is the information that actually comes from your genetic material. This is what your DNA actually encodes, the order of the amino acids. But the order of these amino acids, while being composed entirely of peptide bonds synthesized by ribosomes, is also going to be placing these amino acids in close proximity to each other. And so secondary structure starts to take shape. When you have these nearby acidic and basic groups, these backbone carboxyl and amino groups that are near each other, they're going to start interacting via hydrogen bonding. So this is going to be hydrogen bonding, which is different from primary structure. Primary structure was covalent peptide bonds formed by ribosomes. This hydrogen bonding is not driven by enzymes, but it is going to be due to the close nature of this amino acid backbone. So non-adjacent amino acids will begin to interact. The 2 structures they form are alpha helices and beta sheets. Tertiary structure is as these structures, these secondary structures, bring the amino acids closer together, you'll notice that those variable groups, those R groups or side chains, start to be brought closer together as well. And as you bring these unique variable groups together, you start to really get additional levels of structure. So where the type of structure that was in primary structure was very defined and there was only one component, it was a peptide bond, a covalent bond that was from one carboxyl to one amino group. The secondary structure is a little bit more diverse, and we get hydrogen bonding between non-adjacent amino acids of the backbone. We get alpha sheets or alpha helices or beta sheets. The tertiary structure now involves those side chains, those really variable different groups that are hydrophilic, hydrophobic, acidic, basic, long, short, and suddenly you get an incredible number of different types of interactions. These can be hydrogen bonds, they can be ionic bonds, they can be covalent bonds, they can be hydrophobic interactions, they can be van der Waals. It seems limitless. And this is the real reason which proteins can form so many different structures. Now, in order for us to really call these proteins, because protein really refers to the fully functional form, there's an additional level of structure. And that is when multiple fully formed, so that means they have primary, secondary, and tertiary structure, fully formed components come together, fully formed polypeptides come together in what is known as quaternary structure. Now, when these proteins get exposed to alterations in pH, salt, temperature, or other environmental factors, this can interfere with that structure. And that interference is known as denaturation. So when you denature a protein, you're actually breaking apart its very particular structure. So denatured proteins have no function because they've lost what? Tell me in Socrative. That's correct. They have lost their shape. Now, I'm going to go briefly through another group of these macromolecules here, which are lipids. Lipids are going to be another relatively abundant component of cells because phospholipids, one of the most biologically important lipids, are the backbone for the waterproof membranes that are absolutely essential for your cell. So there are other lipids that are biologically important. Cholesterols form a component of those biological membranes. Fats are used to store energy, and steroids, which can have hormone functions, for example. Let's look first at fats. So these are examples of molecules that store energy, and they really show you a good example of lipid structure. So lipids are composed primarily of glycerol, which is a 3-carbon molecule. Each one of those carbons has a hydroxyl group attached, and then that hydroxyl group is used in the dehydration synthesis that connects a fatty acid onto each of those carbons. So what you get is this 3-carbon backbone with very long hydrocarbon fatty acid tails coming off. The linkage that forms between the fatty acid and that glycerol is known as an ester linkage. It's catalyzed by unique enzymes, and it just depends, quite honestly, on what type of fat and exactly what you're doing as to which enzyme you would be using. I do want to point out, though, that these are, although they use dehydration synthesis, these are not true monomers, or I'm sorry, true polymers, because you're not attaching multiple monomers together. This is 2 different molecules, a glycerol and a fatty acid. So go ahead and tell me in Socrative, if you take a look at this dehydration synthesis, which of the circled groups is the carboxyl group of the fatty acid molecule? Good, the answer is C. Okay, so remember that carboxyl groups are going to be composed of not only carbonyl, but hydroxyl. So you have to have a C doubly bonded to an O, as well as an OH group. Now, fats can vary dramatically. The most important of them is going to be saturated versus unsaturated. Unsaturated fats have a double bond, which means that they have fewer hydrogens, since some of the electrons are being shared carbon to carbon instead of every possible bond being an extra hydrogen. That's why they're called unsaturated. Saturated fats are fully saturated in hydrogens, and unsaturated have fewer. That bending, though, that is caused in that unsaturated fatty acid tail creates an additional shape. And in actuality, that shape is very important. When you have unsaturated fatty acid tails in a phospholipid, they create space between the phospholipids of membranes. That allows for those membranes to become a little bit more liquidy. Now you see this when you look at butter versus olive oil. Butter is going to be solid at room temperature because it's a saturated fat. But unsaturated fats like olive oil are actually going to be liquid at room temperature. Now, phospholipids are going to have 2 fatty acid tails, just like fats do. One of them will be saturated and one will be unsaturated. But that 3rd carbon in the glycerol is going to have a phosphate group attached. You might recall from our function group lecture that that phosphate is hydrophilic. So one side of the molecule will interact readily with water via hydrogen bonding, but those hydrophobic fatty acid tails will not. And so when you place these phospholipids together into water, they will self-assemble into a bilayer called a membrane. That watertight boundary is going to be the membranes that you know that form the boundary around the cell, as well as forming different compartments within the cell. Lecture 5 is going to be all about these membranes.